Hello and welcome. It's 1:02, so we're going to begin. I think a number of people have joined us as well. Happy Wednesday. Today, we're delighted to present a webinar with Ash Dugar, who is the Senior Vice President and Head of Medical Affairs for Dyne Therapeutics. And joined with us as well is Catherine Beaverson, who's the Vice President of Global Patient Advocacy. We're really thrilled to talk about Dyne this afternoon. And for all of you online, please feel free to put your questions in the chat. We will have Ash go through the slides and then we'll come back to questions following his presentation. So with that, I'm going to turn the mic over to Ash. Welcome.
Great, thank you, Pat. As Pat said, my name is Ash Dugar, and I'm the Chief Medical Affairs Officer at Dyne Therapeutics. Before I start, I want to thank Pat, Eric, Emily, and the entire PPMD team to present our most recent data release from the ongoing phase 1/2 study, the DELIVER study, for individuals with 51 skip amenable mutations. It's always a privilege to speak to this community, and I want to thank you for your time and attention today. And as Pat said, with me is the head of patient affairs, someone that you know very, very well, Catherine Beaverson. And we've been together and with this community for a long time, and we're grateful to be here again. Catherine?
Thank you, Ash, and it's great to be with everybody, and thank you, Pat and team, for inviting us to be able to present today. Thank you.
And Catherine will join me at the end for question and answers. So you'll see her again in a little bit. Next slide, please. I'd like to remind everyone that we will be making forward-looking statements today, and you can refer to our website for more information. Next slide. So this slide is very important to me, and you've heard me talk about why this is important in the past. At Dyne, it all starts with our mission to deliver life-transforming therapies for people living with serious muscle diseases, with Duchenne being a priority. This mission is woven deeply into our core values, and it inspires us each and every day. Next slide, please. I'll give you a quick overview of the FORCE platform. This platform was developed to overcome limitations of oligonucleotide delivery to muscle, oligonucleotides such as exon skipping PMOs. So trying to get them into the muscle in a targeted manner where it needs to go with limited or no off-target effects is really what the FORCE platform is designed to do. Now, the platform consists of 3 components. The first is this antigen binding fragment or antibody fragment or Fab that you can see here. Now, we've designed this Fab to be specific and bind tightly to something called the transferrin receptor 1 or TFR1 receptor. And I'll talk more about that in an upcoming slide. Now, this targeting allows for broad distribution of our disease-modifying drugs to tissues relevant to the disease pathology. Now, the choice we made to use a part of the full antibody to TFR1, which you can see in the Y-shaped figure is the full antibody, we've actually taken the top portion, this Fab, and this choice is really important because it doesn't include elements that may lead to greater immunogenicity, and the Fab's smaller size makes it easier to get into the muscle cells as well. Now, the Fab is linked to a payload, and this modularity of the FORCE platform provides the flexibility to be able to send different types of payloads that are rationally selected to target the genetic basis of the disease that we're going after. So, for example, FORCE can deliver antisense oligo payloads such as exon skipping PMOs that need to go to the nucleus of the cell for conditions such as Duchenne. Alternatively, it can deliver other materials, such as something called siRNA, for genetic targets in the cytoplasm of the cell, as is needed to treat something like FSHD, which is another neuromuscular disorder. And really, the goal is to deliver these treatments to the cellular compartments where they can most effectively target the genetic cause of the disease. And this is really a versatile platform. We've also shown that we can deliver payloads aside from antisense oligonucleotides. In fact, FORCE can be leveraged to deliver even enzymes that might treat conditions such as Pompe disease. So this modular approach provides a lot of flexibility in the ability to deploy different targets for genetic disorders. Next slide, please. And as I said, the FORCE platform was designed to overcome this greatest barrier in treatment, which is to efficiently deliver the ASO to where it needs to go with minimizing off-target delivery to the body. So the choice of the Fab targeting TFR1 allows the FORCE platform to essentially leverage the natural biology of the cell. What I mean by that is that transferrin receptor 1 is highly expressed in the body, including on the surface of skeletal muscle, including diaphragm, your breathing muscles, cardiac muscle, as well as smooth muscle like the gut. And so this enables targeted delivery of the ASO to these tissues that are relevant to the disease pathology. And Dyne 251 is designed to leverage the transferrin receptor 1 to deliver an exon 51 skipping PMO to the nucleus of cells in the affected tissues, such as skeletal muscle, cardiac muscle, and even smooth muscle like the gut to increase the production of dystrophin. And the goal is to make a shortened, near full length and functional dystrophin. Next slide, please. So on this slide, I want to provide a brief refresher on some of the preclinical data that gave us great confidence to test this approach in individuals with DMD. And this also helped show us the potential of what might be seen in clinical trials. So in a mouse model of DMD, the MDX mouse, which has a mutation in exon 23, the FORCE platform demonstrated broad delivery of the exon skipping agent to cardiac and skeletal muscle, including the diaphragm, with significantly higher exposure levels than with unconjugated PMO. So when we just delivered the PMO with no mechanism to deliver it, we saw very little get into the diaphragm and heart and quadriceps. But when we used our FORCE platform, you can see that there are very high exposure levels in these key muscles. So this broad distribution allowed for robust and durable restoration of dystrophin expression and of restoration of the muscle architecture. On the top row, you can see the dystrophin, you can see the positive dystrophin positive myofibers that go from a not being there in the vehicle treated group to robust expression in the diaphragm, heart, and quadriceps at 4 weeks, 8 weeks, and even out to 12 weeks in the quadriceps. On the bottom, this red staining is laminin staining, and this is really giving us confidence that we're able to restore the cellular architecture as well. So we see the dystrophin getting to the cell membrane where it needs to get to so that it can have functional benefits, and that we're restoring the cellular architecture on the muscle membrane. Now in the clinic, we can only explore the ability to deliver the PMO to skeletal muscle following administration of Dyne 251. In the next sections, I'll show data that support distribution to skeletal muscle that do translate to early and sustained functional improvement in individuals enrolled in the DELIVER clinical trial. Now, distribution to additional muscle types, such as cardiac and diaphragm, are being studied by looking at cardiac and pulmonary endpoints in the trial, but these data will take longer to emerge. Next slide, please. So very briefly, just to recap this first section, as I mentioned, the FORCE platform was really developed to overcome this huge limitation of the ability to deliver exon skipping drugs to the muscle in conditions like Duchenne. And we harness the natural expression of the TFR1 in muscle as well as in the brain for targeted delivery of a rationally selected payload like an exon skipping PMO with the goal of addressing the underlying cause of disease to ultimately deliver functional improvement to patients. And in our preclinical work, we demonstrated that by leveraging the TFR1, the FORCE platform achieves broad distribution to tissues that are relevant to the disease, including skeletal, respiratory, cardiac, and smooth muscles, as well as CNS. Next slide, please. So as you all know better than I, Duchenne is caused by mutations in the DMD gene, which result in the inability to make functional dystrophin protein. PMO-induced exon skipping can restore the open reading frame of the DMD transcript and enable the translation of an internally shortened and functional dystrophin protein. And production of near normal length dystrophin and distribution across the muscle will be essential to slowing or even stopping progression. Next slide, please. And so when it comes to therapeutic approaches in Duchenne, three key considerations are important to keep in mind for any therapeutic. One is increased dystrophin production. Two, the production of high-quality functional dystrophin. And then finally, broad distribution of the exon skipping PMO to key muscles impacted by the disease, such as skeletal, cardiac, and smooth muscle, as well as the CNS. Now, unfortunately, the only currently approved exon 51 skip therapy in the US results in limited increases in dystrophin protein, and this needs to be given on a weekly basis. Next slide, please. As I said, the quality of dystrophin may also be critical to have functional benefit. Just at a very high level, here we see a milder clinical phenotype in Becker muscular dystrophy individuals who carry mutations that produce dystrophin similar to the skipped dystrophin that would result from exon skipping of similar mutations in people living with DMD. The point here is that dystrophin from exon skipping strategies may have functional benefits and lead to an improved clinical phenotype. Next slide, please. Now, in contrast to exon skip-produced dystrophin, gene therapies produce a much shorter protein called micro or mini dystrophin, which lacks many of the functional domains of full-length dystrophin. There are several different microdystrophin constructs that are either approved or under investigation. And data on safety as well as the durability of effect with microdystrophin gene therapy are still emerging, and it also remains to be seen if long-term functionality is maintained. It is really wonderful to see so many products that are either on the market in the US or that are being investigated for the community. Next slide, please. Lastly, broad and effective distribution of treatments that generate dystrophin to key muscles is also important for optimal outcomes in Duchenne. So not only is skeletal muscle affected in DMD, cardiorespiratory complications are the primary cause of the high morbidity and mortality in individuals. And therefore, therapeutic approaches require broad distribution to cardiac and respiratory tissues as well. Moreover, anywhere from 30 to 50% of individuals with DMD experience neuropsychiatric symptoms. Therefore, the ability to cross the blood-brain barrier and deliver to the CNS may be an important attribute for an effective DMD therapy. Next slide, please. So now I'll transition into talking much more about the DELIVER study and the data. So Dyne 251 is currently being studied in the phase 1/2 DELIVER trial of ambulant and non-ambulant males with Duchenne who are age 4 to 16 years and have mutations amenable to exon 51 skipping therapy. A total of 54 participants have received Dyne 251 across 7 dose and dose regimen cohorts, which you can see on the left-hand side of the slide. And this MAD portion, this multiple ascending dose portion, was also a placebo-controlled, had a placebo-control arm as well. Now, the MAD portion is completed. And so we are very pleased to say that we have now transitioned everyone to our go-forward dose of 20 milligrams per kilogram dosed every 4 weeks, which will be our go-forward dose in the registrational cohort. I will make one note that these doses refer to the exon skipping or the PMO component of Dyne 251. Now, participants treated during the MAD portion of the study had the opportunity, as I said, to transition to 20 mg per kg in the open label extension and long-term extension. And participants who were randomized to receive 40 milligrams per kilogram of Dyne 251 transitioned to 20 milligrams per kilogram in the placebo-controlled period following the review of the safety profile at 40 milligrams per kilogram. And I'll talk more about the safety profile in a moment. For other participants, the transition to the 20 mg per kg dose occurred at different times during their entry into the open label and long-term into the open label extension period. The primary endpoints for this study are safety, tolerability, and change from baseline in dystrophin levels as measured by Western blot. Secondary endpoints include measures of muscle function that you're very familiar with, like NSAA and time to rise and 10-meter walk run, as well as real-world outcomes, such as stride velocity 95C, and I'll talk more about that later. While safety data for all participants in the MAD cohorts will be shown here, I am going to focus on the efficacy data in the 10 and 20 milligram per kilogram cohorts dosed every 4 weeks, and you can see it in the red box on the slide. Now, as DELIVER was designed to be a potentially registrational study, data collected during the MAD portion of the study, as I said, led to the selection of the 20 mg per kg monthly dosing of Dyne 251 as the dose and dose regimen that we're going to go forward with. Next slide, please. Great. So in the registrational expansion cohort, 32 participants have been randomized to receive 20 milligrams per kilogram dosed monthly of Dyne 251 or placebo. We're really pleased to say the cohort is now fully enrolled with data planned for late this year. The primary endpoint, again, is change from baseline in dystrophin protein as measured by Western blot at 6 months. And we have a number of additional biomarker and functional endpoints that are also being studied, which you can see on the right-hand side of the slide. Next slide, please. So I'm going to start to go through some of the data now, but first, these are the baseline characteristics for DELIVER in the 10 and 20 milligram per kilogram cohorts. Now, these cohorts are somewhat similar, or generally similar, with a few notable exceptions. The 20 mg per kg cohort is on average older, with an on average worse function at baseline. And you can see this reflected in its lower NSAA score at baseline, about 10 points lower than the 10 milligram per kilogram arm, slower on the 10-meter walk run, and the stride velocity 95th centile. Therefore, participants in this cohort had more severe disease than in the 10 mg per kg cohort. I will point out that based on the NSAA, 10-meter walk run, and SV95C measures, the baseline time to rise may appear as an outlier between the two cohorts. So we just want to be really clear that participants who could not complete the assessment, the TTR, were excluded from the analysis. And missing times from these participants were not adjusted statistically, an imputation method, which we may look at in the future. Next slide, please. So we believe the true goal of therapies in DMD should be functional benefit. Dyne 251 resulted in long-term functional improvement or stabilization versus baseline across multiple functional endpoints, including NSAA, time to rise, and 10-meter walk run. Now, in the 10 milligram per kilogram cohort, there is a continued and consistent benefit across these functional measures through 18 months. Now, as I mentioned before, upon entering the open label extension, participants on the 10 mg per kg dose were escalated to 20 mg per kg at different times. But at 12 months, all participants were now at 20 milligrams per kilogram. Therefore, the sustained effect primarily reflects treatment at the 20 mg per kg regimen dosed every month. And as I said, that's the selected go-forward dose and DELIVER. Now, the extension period data for this cohort shows the benefit of higher dose and deepening effect on function with longer time on therapy across these measures. Improvement or stabilization versus baseline is also consistently seen at 6 months through 12 months in the 20 milligram per kilogram cohort. And we think this is especially notable given that this cohort was more functionally impaired at baseline. Next slide, please. So as I mentioned before, we have an additional functional assessment in DELIVER called the stride velocity 95th centile. It is a digital and objective continuous measure of ambulation in the patient's normal daily setting. It's been shown to correlate with traditional ambulatory measures such as NSAA and 6-meter walk test. And it has been validated as a primary endpoint in Duchenne clinical studies by the European Medicines Agency, essentially the equivalent of the FDA. The SV95C demonstrated greater sensitivity as well, meaning that it can detect change earlier versus other motor function tests. And in the validation work that's been done around this particular measure, it's really important to note that a minimally clinically important difference in this measure is about 0.1 meters per second. And so this helps us, and this corresponds to about a 30-meter change on 6-meter walk test. So I bring this up because this is a really robust measure that's been correlated with measures that are well understood, like 6-minute walk and NSAA. Next slide, please. So when we look at this data, Dyne 251 led to early and long-term functional improvement in SV95C at both the 10 and 20 mg per kg cohorts. Improvements versus baseline are sustained through month 12 for the 20 mg per kg cohort and month 18 for the cohort that started at 10 milligrams per kilogram. And again, you can see here that at month 12, everyone in the 10 milligram per kilogram cohort transitioned to 20 milligrams per kilogram. Really important, the change from baseline in SV95C following treatment with Dyne 251 achieved and exceeded the proposed minimally clinically important difference by 6 months, thereby reflecting early and meaningful functional improvement with Dyne 251. So it's always one thing to see something that might be statistically different, but is it clinically meaningful? Is it clinically relevant? Does it matter to the individuals living with the condition? So the totality of functional data shows that treatment with Dyne 251 consistently results in early and sustained functional benefit across multiple motor function assessments and across different levels of baseline function. So regardless of where they're starting in terms of baseline disorder, baseline disease, we're able to show benefit or stabilization. Next slide, please. So lastly, the functional improvement with Dyne 251 is driven by the dystrophin produced. We talked about quantity, quality, and distribution. And this slide shows dystrophin analysis as measured by Western blot for both unadjusted and adjusted for muscle content at 6 months. At the 20 milligram per kilogram dose, Dyne 251 resulted in 3.7% normal dystrophin, and that's the unadjusted. When we did adjust for muscle content, and you can see mathematically how we calculated that, we really feel that it's important to be transparent about how we are coming up with the muscle content adjusted dystrophin number. And when we applied this mathematical formula, we show about an 8.7% dystrophin level in the 20 milligram per kilogram arm. Next slide, please. So Dyne 251 demonstrated a continued favorable safety profile with the majority of treatment-emergent adverse events reported as mild or moderate. Now, substantial safety data has been collected in DELIVER with total doses of administered study drug approaching 1,000 over 77 patient years of follow-up, with some participants on study for nearly 2.5 years. These data also reflect over 500 doses of study drug administered at the registrational go-forward 20 milligram per kilogram dose. I do want to note that 3 serious adverse events potentially related to study drug occurred in 2 participants in the 40 milligram per kilogram cohort. These have been previously reported and characterized, and both participants have fully recovered. There have been no other safety signals of note. So outside of the 2 participants with those drug-related serious adverse events, no other participants had, we've not seen other SAEs. And importantly, no other participants have had persistent anemia or thrombocytopenia. And there have also been no clinically meaningful electrolyte changes, such as with magnesium or potassium. Next slide, please. So this data set from Deliver illustrates the promise of the FORCE platform in developing some next generation exon skipping therapies and supports pipeline expansion. And so we have preclinical programs that are targeting additional exons, including exons 53, 45, and 44. And I look forward to the opportunity to update the community on the progress for those programs. Next slide, please. So this is a busy slide. I'm not going to go through this point by point, but maybe I'll just highlight a few things that, what we were able to demonstrate in the preclinical models did translate into what we are seeing in the clinic so far. We're really pleased to be able to see the levels of dystrophin restoration, and more importantly, what we are seeing in terms of the totality of evidence with the functional benefits that we're seeing in the study at multiple cohorts, even with differences in terms of where patients are starting in baseline disease. And we're seeing improvements and stabilization that is divergent from natural history and also above, in some cases above, what is deemed minimally clinically important from the literature. Very importantly, we anticipate filing our application for submission for US accelerated approval early next year. And we're really pleased that we were granted orphan drug and rare pediatric disease designations in March of this year. And we look forward to continuing to expand our portfolio. Next slide, please. Great. So I think one critical aspect of the multi-system nature of Duchenne that for a long time has gone overlooked in research is the brain involvement in Duchenne. And of recent, thanks to the amazing work of so many in the field, of the BIND consortium and many other efforts, more and more research is being done in this area. And there's been recognition that of some core areas of brain involvement identified as particularly affecting individuals living with DMD. So these include neurocognitive issues where people often experience mild to moderate intellectual disabilities with particular difficulties in verbal IQ, working memory, and executive functioning. Academic and learning challenges are common, especially in reading, dyslexia-like symptoms, and arithmetic, and this has significant impact on school performance. Increased risk of anxiety and depression is observed and often linked to social difficulties, frustrations with physical limitations, and progressive loss of independence. And neuropsychiatric issues are often seen. This includes higher prevalence of autism spectrum disorder, ADHD, and obsessive compulsive disorder, likely due to dystrophin-related brain dysfunction, affecting some of the synaptic signaling and connectivity. Next slide, please. So we wanted to explore whether or not the FORCE platform might have the potential to address some of these symptoms. So this slide shows that the FORCE platform delivers our exon skipping PMO and induces exon skipping in the brain of a DMD mouse. So again, given the pervasiveness of neurocognitive manifestations in individuals with DMD, and knowing that TFR1 is expressed in the brain, we wanted to test if the FORCE platform could deliver payload to the CNS to potentially address some of these symptoms. And what we used in this study is something called an HTFR1 MDX mouse model. And just what I mean by that is, the MDX mouse model that's been used by us and many others, which is that exon 23 mutation. Well, what we've done is created a mouse model that expresses the human TFR1 receptor. And this is really important. So that transferrin receptor on this mouse model recognizes the human version of that antibody. So here in the top row, you can see that 7 days after administration, a single intravenous dose of our conjugate, but not the unconjugated version, or we have a negative control as well, which I won't get into here. But essentially what we're seeing is that the FORCE platform versus something delivering the exon 23 PMO without any conjugation is really showing a big differential in terms of getting into key areas of the brain, such as the cortex, the hippocampus, and the cerebellum. The bottom row shows that the bottom graphs show that this widespread PMO distribution in the CNS resulted in the target engagement as demonstrated by exon skipping. Next slide, please. So the effective PMO delivery and exon skipping were maintained 28 days after the administration of a single forced dose in that cortex, cerebellum, and the deep brain. And what I mean by deep brain, it's kind of a catch-all term for the anatomical region that's likely to contain very small parts of the brain, like the amygdala, that have a huge impact on brain function. And also, this effective PMO delivery and exon skipping in the CNS did restore dystrophin expression, as seen on the graph on the right-hand side. Next slide, please. So as you look at this slide, I'll sort of talk through a few things. What about the impact of our force conjugate CNS delivery on the phenotype, on what's actually happening behaviorally? So it's known that MDX mice exhibit something called anxiety-induced freezing behavior in response to restraint. So we were able to show that in this MDX mouse that expresses the human transferrin receptor, the same thing occurs. And this restraint-induced freezing is assessed by scruffing, or just grabbing the mice by the skin of their neck for 15 or 20 seconds. So this causes the mouse to freeze. And so what you can see here is this video is playing on a loop on the left-hand side. And the mouse that's treated with vehicle on the top left panel is not moving. And you'll see it again right here. So that mouse was scruffed and is not moving. Now, the bottom video shows what happens in the presence of force. This mouse is moving. So the graph on the right shows that a single dose of force can lead to a complete and a nearly complete and durable resolution of up to 2 months post-dose of this anxiety-induced freezing behavior in the HTFR1 MDX mouse. I know this is probably some new type of information that we're showing here, so happy to take questions at the end to go into more detail or explain this further. Next slide, please. So just very, very briefly, just to recap some of the key points. As I mentioned, our platform leverages the natural expression of TFR1. And one thing I failed to mention is the function of TFR1 is to maintain iron homeostasis. It's involved in iron transport, which your muscle cells need. So again, we are using the body's natural cell biology to deliver our exon skipping PMO. We've shown that Dyne 251 has a favorable safety profile, with some participants on therapy for 2.5 years. And we've been able to show long-term improvement in measures of clinical and real-world functional outcomes in individuals with Duchenne. We believe the FORCE platform has the potential to transform lives of individuals living with these neuromuscular disorders by addressing the totality of symptoms experienced by these individuals, including CNS manifestations. Next slide, please. And so, as I end, I just want to say thank you to the families, investigators, the site staff that have made this study and so many other advancements in Duchenne possible. We know that over decades, there have been countless sacrifices, and it's only because of your dedication that we're able to actually advance therapeutics that will hopefully bear fruit. And what you've all done over the years is nothing short of extraordinary. Next slide, please. So, with that, I just want to, again, thank the entire PPMD organization for the opportunity to present to you today. And if you'll indulge me for a moment, I just want to say to Pat and team how incredibly grateful I am for the nearly 10 years that you've been teaching me and countless others about Duchenne and its impact on families, so that we can, in turn, try to contribute to eliminating this condition. So thank you all very much.
Thank you, Ash. That was very, very helpful. And I so appreciate you and Catherine joining us today. I think just because of the interest of time, I'm going to go backwards through the slide. Usually I start at the beginning and go through, but I think there are some really practical questions I would like to ask you and Catherine at the beginning so that we can go backward. And so Emily, if you'll go back to the previous slide and we'll just go sequentially back. You have done a global study, which is something we as a community are quite used to and thrilled about. What I think is worrisome to the people who are outside the United States, in terms of, you mentioned in an earlier slide that you will be submitting the package in hopes of an accelerated approval, which means if it's in early 26, that whole process is about once the package is submitted, 8 months, 60 days for the FDA to agree to review, and then 6 more months before a decision occurs.
Yeah.
So the people that are in other countries, is Dyne committed to provide this this therapy to the individuals who participated, and for how long? How is that looking, so that we in the US, if I cross fingers so that we have an approval here and then a greater number of people will have access to the therapy. But what about those people that have given blood, sweat, and tears into other stuff, into outside the US?
Yeah, no, it's a really, really important question. Thank you. So I want to be very, very clear that anyone who has participated in our study will continue to get drug. We are committed to making sure that, upon the completion of the DELIVER study, if there is not marketing authorization at the time, or there's some time before we're able to get approval outside of the US, that every patient who's been on DELIVER will continue to receive drug if they so choose. We will not be leaving anybody without drug who has been in our trial. And then just maybe taking it maybe even a little broader, we are not only looking at expedited approval in the US, but we are also having discussions with agencies outside of the US for expedited approvals for Dyne 251. And so we are, not just saying, okay, it's the US and accelerated approval, but we're trying to bring this to patients as soon as possible all over the world.
Thank you, Ash. So that means whatever country you're in, you would provide access until there's access that is through the system that exists in those countries.
Exactly.
Okay, back up one more slide, Emily. Maybe one more, sorry. I think this is amazing that you have some initial evidence that this therapy does cross into the blood-brain barrier. I know that all of us have seen children on the spectrum with behaviors that are on the spectrum and or some impairments in the amygdala, the frontal cortex, etc. So I wonder, and this is perhaps something you can't answer, but my question about this, Ash and Catherine, is that the children, that I always say children, but I'm old enough to call everyone children, even if they're adults. So I apologize for that right off. We know that steroids exacerbate some of these behaviors, right? And so I wonder, when you were treating these mice, were they on steroids as well, so that we could get the full brunt? Because as I understand, steroids are standard of care. They're not likely to go away anytime soon, as far as I know. And so I wonder if the steroid impact has been evaluated in light of the therapy.
Yeah, great question. So these mice were not on a background of steroids. And I'm probably less than qualified, but I'm not certain about how much a background of steroids would then help us really understand the CNS benefits. I don't know what the impact of chronic steroid use would be on the MDX mice. So it's something that we can dig into and come back to the community with.
Thank you. We are holding a brain meeting in the fall, so maybe we can raise that as well, because I think it's an interesting question. Which brings me to another sort of clinical question, and this is an overall clinical question of I think, as you know, steroids are standard of care, but they're also sort of our worst nightmare in many respects, because of the impact they have on bone and growth and et cetera. And so two questions. One is, in the middle of this study, based on the access to vamorolone, can people switch to vamorolone should they wish to? Can they switch to a different regimen of steroids? And do you have any thoughts on or willingness for that switch or that change? And then in light of the therapy, would there be, in your estimation, a time when we could avoid all steroids? So steroid changes, steroid changes in regimen, changes in steroid, the actual Agamree versus deflazacort or prednisone
Right.
during the study, and then ultimately, could we get off of steroids? Do you imagine a day for that?
Yeah. Wow. So let me try to answer the first part, which is probably a little easier. So within Deliver, we do require when you enter the study that you have been on a stable dose and dose regimen for the previous 12 weeks. The adjustments can only be because of weight. So one cannot, for example, in the open label extension, one cannot go from deflazacort to vamorolone. So that is not allowed within Deliver. I think that once we have seen marketing authorization in different jurisdictions, we then have updated the ability to include vamorolone in patients coming in new into the study. So once it was approved in the US, we then said, okay, now patients who are on vamorolone can come into the study, but you cannot switch from one steroid to another in the open label extension for Deliver.
Switch to a regimen from daily to weekend.
And you cannot switch regimens either. Exactly. So you have to stay on that same dose, the same drug, and then the same regimen, and then adjustments on dose are only due to weight. And then on the steroid, and I'll probably be very brief here, and I'd ask Catherine to please chime in too. On steroid sparing, I think that would be tremendous if we could find find this therapy or other therapies where the need for steroids are eliminated. Obviously, I think that would be an incredible thing. We are planning in our phase 3, again, though, to be adding or to have Dyne 251 on top of steroids. So right now, we recognize that steroids are standard of care in the US and many other jurisdictions. And so we are looking at our drug on top of steroids. We would love to be able to have a time when steroids could be eliminated, frankly. And I don't know, we've talked a lot about this in different form, as you know, if you start very, very early in a 3, 4, 5-year-old, then can you eliminate the need for steroids if you start that early? I think there's a lot of data that has to be generated and a lot more discussions as well. But right now it's standard of care, so we add it on top. Catherine, do you want to add?
Great, thank
Thanks,
you.
Asha. Nothing to add. Thank you.
Great, Emily, if you'll back up to the SV95C, I think that might be 1 or 2 slides back. Maybe just, yeah, another slide. Maybe let's go to the expression data. Another slide back. Another one.
And my apologies, Pat, for not having slide numbers on here.
No, I think it was, right here. So let's stop here and just go back sequentially. You might imagine that we're all anxious to see, the rest of these exons come forward. I see that they're going into preclinical, which means they're one step closer to getting in the clinic. I'm guessing you are not able to tell us a timeline for those exons, but they're moving forward at the same. So I guess what I really like to nail down is, as you're thinking of 53, 45, and 44, do you anticipate rolling them out at the same time or one by one by one? And is there something in terms of a platform where you could think about a study where all 3 of these would come at the same time?
Yeah. And so I think you're thinking along the same lines that we've been thinking about it. So these candidates are moving forward in parallel. We also know that the FDA has recently come out with their platform technologies guidance, which opens up the door to be able to being able to actually approach these in a platform approach, where you can basket these candidates, run a trial in parallel with hopefully just no placebo arm or one placebo arm after you've shown the benefit of another compound using the same platform, in this case, like Dyne 251. So we are looking to engage with the FDA to leverage the platform technologies guidance, as many other companies are as well, which I think is really, really important. But we'd love to be able to find an approach with regulators to do exactly what you said, move these in parallel in a basket study. Otherwise, what we've seen right in the past is it's 4 years to develop one, 4 years for the next one, 4 years for the next one, 4 years with the next one, when you need sort of full non-clinical and clinical data packages. So hopefully we can leverage the Dyne 251. There's a couple of things for Dyne. One is we're developing other drugs with the same platform. So we can leverage that safety, that accumulation of safety data with the platform to help with that argument. Then we're going to hopefully demonstrate the benefit with Dyne 251, use that evidence as well to be able to talk to the FDA about this platform approach. So that is definitely ideal. We are committed to advancing these other exons, and we're trying to find the most efficient path to do that.
And Pat, as a follow-up to your question, may I just ask, Ash, you mentioned the draft guidance for the platform technology designation. Has there been a final guidance put forward?
No, Catherine, there hasn't been a final guidance put forward on platform. And as you know, the language in that guidance is really, I'll say loose, and in terms of it's not really definitive about you have to do A, B, and C. I recall back in 2010 when the NIH and FDA had a meeting and we talked about antisense oligonucleotides as a general vehicle for possible therapeutics. And at that time, the position of both of FDA and NIH was that they wanted to see one of the exons go through the whole phase 1 through 3 and get approved, and then they would be interested in or willing to talk about a platform approach to the rest. I think this is something advocacy needs to work on, Catherine, and we're certainly going to pull you in on trying to think through that. I do know that with the new administration, Dr. Makari and Dr. Prasad, I know that they're having open discussions in terms of entertaining a lot of different voices. So certainly our voice will be in there. I also know the next PDUFA is coming up in a couple of years, and I think this is where advocacy can really enter in and try to get this a little more clear, both in legislation as well as in an updated guidance by FDA. So I think this is where we have to go because time, the coin of phrase, time is muscle, and people waiting on these other exons just feels heartbreaking for all of us, which brings me to the rare exons. And I know that's on your mind and we won't go through all the rare exons, but I think, because when the first antisense therapy was approved in 2016, there was this belief that you could treat about 80 plus or minus % of this population with an antisense oligonucleotide. And I think those 4 exons would treat, I'm guessing, a little bit more than 45%. So I think we need to get to the rest of those exons as quickly as possible. So I thank you for that. If you'll go back another slide, Emily, that would be great. Just a question here, Ash, on the most frequent treatment AEs that I see, there's pyrexia, and this is generalized itching. Is this, and it's 48%, are all the boys or many of the boys, 48% of them having itching? How long does it, having the pyrexia, does it last a long time? Is it only during the infusion, and are you treating it prior?
Yeah, so in terms of, I think, for some of these cases, we haven't seen like a specific pattern of the pyrexia. We're seeing that they're having, increased temperature, for example. So we're not seeing a consistent pattern where it's like 2 hours after infusion or 2 days or what have you. It could be after the 4th dose or after the 5th dose, and there's no specific pattern. What I will offer is the feedback from the investigators and, of course, discussions with our own safety group. This has all been very manageable. So whether it's like giving Tylenol beforehand or giving a Tylenol afterwards, but the pyrexia is frequent, as you can see here, but highly manageable, not unusual for drugs that are received in this manner. But we do recognize that nearly half these boys are having this issue. But all of the PIs are able to manage it very, very, very easily.
Thank you. Do you ever give an antihistamine prior to?
Sometimes, yep. That's right. Antihistamines, Benadryl, Tylenol. It really depends on the investigator's judgment.
Okay. Either of those agents seems to take care of the problem. All right, Emily, back up one more. This is the expression data. Ash, I think this is, and Catherine, this is really complicated in terms of adjusted and unadjusted. And I will also say that, you know, sometimes, and you mentioned Becker patients who have an in-frame mutation adding 51, or you know, you're seeing a different natural history there. So can you talk about adjusted and unadjusted and what that really means? And our heads always go when we see 8.2, as we've all heard in this community, that if you're above 5%, that's a Becker phenotype. Can you just give a little color here to adjusted, unadjusted, and does it or does it not relate to a Becker phenotype?
Sure. So I think I'm going to probably separate out the two a little bit. So in terms of the unadjusted versus muscle content adjusted, when we take the biopsies of the bicep, what we're doing is looking at within that muscle biopsy, how much of that muscle is fibrotic. And we're essentially saying, okay, we will only look at the viable muscle portion and do this calculation of dystrophin over percent muscle content to come up with the muscle content adjusted. So really it's just correcting for the amount of fibrosis in that biopsy that we're taking. I think when you know, and perhaps I didn't do a good job of explaining it, but in terms of thinking about the individuals living with Becker's, really it was just, you know, in no way are we implying that we are just trying to convert DMD individuals to Becker's. That is not the goal. And that is not, you know, that's something that, you know, I think has been hopefully, you know, sort of out of the dialogue now for several years. So that's not the goal, but it's really to understand, well, you know, in some of these sort of individuals that are producing dystrophin, but a shortened form of dystrophin, what is their phenotype? And so trying to just think about, okay, if we can actually produce a shortened form of dystrophin, is it going to be functional? And so at the end of the day, what's really important is the functional assessments that we're doing. So dystrophin sort of underpins, right, the biological explanation of the function that we're seeing. But we're trying to take clues from all of the literature that's out there in terms of the importance of exon skipped protein.
Right. I think you're right, that language of Becker, but I think our heads go to Becker frequently with percentage of dystrophin expression. And I think that's sort of is somehow connected in our brains as we hear about the diagnosis. As we're thinking about this, did this, as you looked at this dystrophin expression, did you also see a change in the CK level in these patients, and are you tracking it?
Yeah, we are. So we are tracking the CK. We haven't disclosed that data. We're still accumulating that data and trying to make sure we interpret it appropriately. So I do think at some point we'll be sharing the full data set with the community. But we recognize the CK levels are of high interest as well.
Great, thank you. Let's go backward again, Emily. The SV95C. First of all, I think this is a really amazing device, and we've learned a lot about it, and I'm very happy it's going through the qualification process, which is long in
Yes.
FDA. I wish it would be a little more quickly. The EMA has certainly responded in a much more rapid approach. So you talked about this, a couple questions here. You've gone out to 18 months, and it looks like these patients are stable. Would you expect continued with additional expression over time? Do you feel that stability would be what we're looking at? Do you think with continued expression, we might see an increase in terms of, and last but not least, and I know this is a lot of questions, respectful of your time, you talked about clinically meaningful. Can you talk about how this relates to clinically meaningful benefit?
Sure. So I just want to be careful about not over promising. I recognize that these are still relatively small sample sizes. So I just want to be careful about speculating that we'll see continued improvement at 20 milligrams per kilogram. I think again the part of it depends on age and baseline function, right? As we've been educated by many of the key opinion leaders in the community. So we're really pleased to see stabilization. What we've learned from you, from others, is that if we can stabilize the condition, that is a win. And if we can see continued functional improvement at one or both doses over time, that's going to be hugely beneficial. So, we're excited about what the 20 milligram per kilogram cohort the group that started at 20 mg per kg, what they're going to look like at 18 months and at 24 months. And hopefully we continue to see that trend going up. I will also point out the gray bar is placebo. And so I think, I wouldn't underestimate that difference between treatment and placebo will continue to increase. I feel pretty confident about saying that if those patients were not transitioned in the open label extension to active drug, you would continue to see that decline. And then finally on the MCID, just again very briefly, the validation work for this, the validation work that went in the qualification document is incredibly robust. This is correlated, so for example, to a 30-meter change on 6-MWT. And so this is correlated to things that like NSAA and 6-meter walk, the 6-minute walk. So we feel like that work has shown that there's about a 0.1 MCID that's been identified. And we're able to see something in the realm of 0.1619. So we're really pleased to see a really robust change from baseline and what will likely be a very big separation from placebo. That gives us confidence that this real world measure is assessing something that's truly meaningful in the lives of individuals.
And unbiased, and I think that's a very good way to really assess, because we all sometimes these outcome measures include, like the North Star is influenced by the shoes you're wearing, the nutrition you had, if mom or dad's there, and etc. So I think that this is really a very nice, unbiased, and very helpful way to assess.
And I'll just add to your point. This is a continuous measured over about 4 weeks. So, it takes into account sort of those ebbs and flows of how folks are feeling and the environment. So it's a continuous measure over a period of 3 or 4 weeks.
So Ash, you also have, obviously in an earlier slide talked about the 40 milligram dose, and then you changed everyone down to 20 at the 12-month mark. Is there any reason or rationale for going back up? So some people would ask the question, and I certainly would, if you went to 40, are you going to get greater expression, better response on the SV95C, and overall better? So are you thinking of escalating, or is this the dose that you feel is, and the MCID, maybe you want to explain the minimal clinically effective dose. So
sure. So we saw those 3 SAEs at the 40 milligram per kilogram dose, and out of an abundance of caution, we converted everybody who was on 40 mgs to 20 mgs per kg. So that was the impetus to do that. And what we're seeing is, we believe, very, very good benefit at 20 mgs per kg. Now, you're right. We can go to 30. We can go to 40. We can explore higher doses again. But that would also push out the ability for us to get this agent into the hands of physicians and families. And so I think we want to be thoughtful about balancing, trying to get, which just in drug development is sort of this holy grail of like this perfect dose and dose regimen, which you can explore dose and dose regimen for years, frankly. But we've made, I think, a really good decision that we're showing very good benefit risk at 20 mg per kg. We have the ability to get this filed early next year and get it in the hands of physicians and families. And we can explore higher dose and dose regimens in the post-marketing setting. So there's always that opportunity as well to explore higher doses post-marketing. But we didn't want to delay 6 months or a year or 2 years to continue exploring higher and higher doses.
No, I don't disagree. And just while we're talking stability, a shout out for Eric Camino from PPMD, who's leading a preference study on stability, because what we want is to be able to provide to the FDA is stability is benefit in Duchenne. So I think, I always say, I don't know of any of us in this community who don't go to bed at night and wish everything would obviously improve, but if rather than improve, if we can have stability, that's also a great gift in terms of where a patient is in this disease process. Okay, back up again, Emily. And I know we're a little over time, so if Ash and Catherine, do you have a few more minutes? Can we continue?
Yeah, of course.
Acceptable? Okay, all right. And just one comment here on the SV95C, or I guess two. This is for typically an ambulatory population. I know that they are looking at this in a non-ambulatory population. So did everyone use the SV95C or just the subset who were ambulatory in this study?
So yeah, great point. So the side device, which helps us evaluate this particular outcomes measure, actually collects data for those who are non-ambulant as well. So everyone who was in the 10 mg per kg cohort and 20 mg per kg cohort. So starting at 10 is when we implemented this measure. So all of those individuals have been using the side device. So we are collecting data that's beyond just SV95C. So for those individuals who are non-ambulant, who were in those cohorts, they are also using the side device to collect data.
Thank you. Back again, Emily. So you're collecting multiple endpoints here, and Ash and Catherine, you have in your age range 6 to 16, you have people, I'm guessing, that are no longer ambulatory. So what I didn't see here is a pulmonary outcome, and you have talked about the fact that you are reaching the diaphragm, which is critical here for all of us and for all of these individuals in the study. Can you tell us about, are you collecting pulmonary outcomes, and if so, what are they?
Yeah, no. So yeah, I failed to mention that. So yeah, so we're looking at 4 to 16-year-olds, and to your point, we're looking at FVC percent predicted, PEF. So we are using we are obviously using a spirometer to collect pulmonary function tests, and so we will be providing that data as well at some point. I would also offer, just to your point, we also have cardiac MRI. So we are also collecting cardiac function data in this study, and the PUL 2.0, so performance of our upper limb function as well. So we do recognize, because of that big span, and also that we have transitioning and non-ambulatory patients, we want to collect pulmonary, upper limb function, and cardiac function.
Thank you. I'm really pleased about that, because I think Catherine was in Washington with us 2 years ago when one of the young men in the panel said, improving the pulmonary function or improving respiratory effort by even 5% would enable him to be much more social and not have to take the non-invasive ventilation. So, I mean, I think that incremental benefit is just so impactful on the activities of daily living, and I think that's really terrific that you're reaching all of these tissues to include smooth muscle, which, as you know, we know that there's impairment in smooth muscle over time, but we really haven't studied smooth muscle. So it'll be very interesting to see where we're going here and how much protection we get. So if you'll back up again.
And Pat, to your point, in phase 3, I don't have the ability to give specifics now, but in phase 3, we do plan to implement patient-reported outcomes to look at, issues with gut function. And also, as I was mentioning before, we'll think about CNS measures as well in phase 3.
Right. So we're updating, Rachel's leading the effort to update the standards of care. So we have a GIGU component. So we certainly will be able to contribute to at least some opinions about what sorts of outcomes that we think would be useful. In addition to that, I mentioned we're having a brain meeting. Clearly, the Dyne Project is developing tools, and Molly Colvin and Natalie Truba from Nationwide and Boston, respectively, have developed the BELLS tool. So I wonder, as you're thinking about this cognitive evaluation, that you might be able or be willing to include one of those tools in the phase 3. While we're on the phase 3, is this a placebo-controlled trial? Do you have a protocol? Do you have a timeline when you're thinking about beginning to enroll in that study?
Yeah. So we do have, we've done a lot of thinking around that phase 3 design. As you would imagine, we are having conversations with both regulators, KOLs, and the patient community. So we are, we're not ready to roll out details just yet, but it will be a placebo-controlled phase 3 study as a confirmatory study in the US, as you would expect. And also by regulation, the FDA really wants to see that confirmatory study, sort of initiated at the time of BLA filing. So that helps you understand kind of timing for initiation of phase 3 based on what I was saying about the BLA filing early next year.
Yeah, so we'll be very anxious to see that. We're not really happy about a phase 3 with a placebo controlled. So if you could squish that into the shortest amount possible time, we would certainly appreciate that a lot, because as you know, that's hard. And do you expect a global study as well on the 51 phase 3?
Yes, for sure.
Okay.
And it will
All right.
enroll new patients. We're not taking patients from DELIVER into phase 3, so it's going to enroll new patients.
Okay. In light of that, in terms of your open label extension, are going to continue through that. And I guess one of my questions here, Ash and Catherine, is, are you continuing to collect outcomes on these individuals who were in the first 1-2 study, so that we know over time, or can get some sense of the impact over time and the cumulative evidence of dystrophin expression over, now you've gone to 18 months and beyond, so that we can see over a period of years what that actually occurs?
So absolutely right. We have this DELIVER study that's ongoing for at least 5 years, and the right thing to do is collect long-term outcomes, right? That's just the right thing to do. But second, it just makes sense because we're going to want to be able to compare this to a matched natural history to support discussions with regulators and payers. So we're going to continue to collect outcomes over the duration of the study. We are not going to be collecting necessarily as required by the study multiple biopsies. We have the baseline in 6 months, but we are not right now asking families to come back every year for another biopsy. So we will have very limited ability to show what the dystrophin expression gets to over a long period of time. Perhaps families may opt in if they choose, but that is not part of what DELIVER requires right now.
For the patients in these studies, Ash and Catherine, are you planning to return data to the patients? I think one of the questions will be, how much expression did I or did my son experience with the therapy? Are you willing to disclose or planning to disclose at any point in time?
Catherine, do you want to take that? Do you want me to take that?
No, that $1 million question goes to you, Ash.
Okay, great. So we've talked a lot about this internally, and I feel like that's the same answer I give you every time you ask me. We have to figure out the mechanism to give data back. I think we feel very strongly that there are models out there that you've shown us, right? That you guys have taught us, but there are models out there for data sharing. There's been like one or two really good examples in the Duchenne space of giving data back to families. So our goal is to give data back to families at a time when it does not jeopardize the ultimate approval, because that'll just do more harm than good. But we do believe there's a need to share the data, whether it's placebo data, baseline data, or what have you, with the families and with the community. We just need to figure out what that mechanism looks like. But there are models that are out there for sure.
Yeah, I think we're all curious, and as I've said before, restoring dystrophin feels like foundational, at least in our experience and our preference data. We know that is sort of the holy grail, if you will, or the preference to get dystrophin expressed and build on top of it, or build before it and then get it expressed, either way. But I think having that understanding of what was expressed and the cumulative evidence of benefit would be great. Just a couple questions. I know we're really over time. But and probably too, when we were talking about the pyrexia following that, there's a question: did we confuse pyrexia and pruritus?
Well, the pyrexia was the high temperature; it was the fever.
Yeah,
but I know what you're saying. I
yeah,
think so. Just to be really clear, the pyrexia
I
is
did confuse pyrexia with pruritus. I got to get back into nursing and figure it out, right, and get my terms. So I apologize for that. One of the questions was, do you think there will be any resistance to dystrophin expression over time?
Resistance to dystrophin expression over time. Do they mean like anti-drug antibodies?
Yeah, like
No, like you would build
it-
resistance to the drug and you would no longer express.
exactly. Thanks, Dan.
there's no, I mean, I'm trying to think about a mechanistic reason for why that would occur. I don't believe there's any reason for that to occur. What we're seeing, of course, is 6-month dystrophin, but we're seeing functional benefit that lasts out to 18 months. So I don't believe there's no reason to believe that there's going to be that occurring in the study.
Yeah.
But I may be
And I would agree,
underthinking this.
since the 16 PMOs were approved 2016, we have not seen or heard any SE or any effects from that or negative effects. So I wouldn't suspect that either. And one of the last questions was in the phase 3, these will be all new patients, not existing patients that transfer back. And then finally, should it be approved, can we just go from whatever 51 skip therapy a person's on to Dyne 251? Would there be any reason that we couldn't switch?
Yeah, no. I mean, in the trial, we require a 12-week washout, but that's really for trial purposes. But once approved, our position is that there's no reason that individuals who are amenable to 51 skipping who are on other drugs, like ETAP or some of the individuals who are amenable who might even be on golodirsen, they could switch to Dyne 251.
If a patient was, if once this
And we're also dosing every month. Again, as I said before, we're dosing every month. Yeah.
and once this therapy is approved, or even during a study, if a patient was on cardiac drugs, ICE inhibitors, and ARB, losartan is the ARB, other drugs such as Entresto, would that be contraindicated in the study? And would it be contraindicated as the drug is approved?
Yeah, so definitely not contraindicated in the study. So we recognize that individuals coming into the study are on various cardiac medications, and so that is allowed in the study as per judgment of the physician and the PI. So again, if there's any, what's not allowed except for specific reasons are like huge changes in cardiac management, but absolutely allowed. And then in clinical practice, again, that would be perfectly fine.
And then if an individual has been involved in a gene therapy protocol or received the approved product in the US, Elevidys, will those individuals be allowed in this phase 3 study?
Great question. No. So those who are on previous gene therapy currently will not be allowed in the phase 3. And I want to be just really clear. We're still evaluating this. So I'm providing you some preliminary thoughts, but we still need to think about this. And we also know realistically there's a world in which families will want to be on anything that can provide benefit, gene therapy, exon skipping, givinostat, etc. And so, it's incumbent upon Dyne and many other sponsors to generate data that actually provides information for physicians and for payers. And I think, that is an important consideration too, in terms of what the payers will require for allowing multiple treatments that are approved.
Yeah, Ash, I think at least when early days of PPMD, we talked about a cocktail of therapies, and we're getting the therapies in terms of we're lucky enough in the US to have 8 approved therapies and another pending in August. I think how best to combine them in what sequence for what patient, and to collect the data so that the payers are able to cover those therapies is going to be critically important. So I thank you. So likewise, if you're on givinostat, will that be included in this phase 3?
Again, I think we just have more discussions to have internally around inclusion exclusion with respect to givinostat. So once we are more solidly placed in providing kind of the details on phase 3, we will definitely do that. But we're having all of these discussions internally right now.
We'll look forward to hearing about the phase 3. We are now 15 minutes over time.
Sorry.
I can't thank you enough for doing this. We're very enthusiastic about the FORCE platform, not only what it can do for all of these individuals who are could be benefited by an exon skipping therapy in Duchenne, but certainly the broader range of patients who need and deserve help as well. Catherine, thank you, Ash. Thank you, Catherine. Thank you, Ash, for always joining us and always answering the 1,000 questions we have. I'm sure we're not at the end of questions, but we'll look forward to seeing you at the conference soon in Las Vegas. And then we'll see you soon again in a webinar. Thank you all for joining us today. I hope you found this useful. Please contact us with additional questions and we'll see that they're answered. And really have a nice weekend coming up. Thank you again, Ash and Catherine.
Thank you.
Thank you.